side of the globe. Very excited and thrilled to welcome you all to the first GKII webinar of 2023. I'm Priyanka Das, the Program Manager for GKII and your host for today's webinar. Our distinguished speaker for today is Dr. Sheela Magge. Welcome, Dr. Sheela.
Thank you.
She is the Director of the Division of Pediatric Endocrinology and the Lawson Wilkins, with Endowed Chair of Pediatric Endocrinology and the Associate Professor of Pediatrics at the Johns Hopkins School of Medicine. Dr. Sheela's clinical interests include all endocrine disorders in children and adolescents, particularly type 2 diabetes mellitus, prediabetes, obesity, congenital insulin resistance syndromes, amongst others. Dr. Sheela is also an eminent researcher. To name a few, she is currently co-principal investigator of an NIH-NICHD R01 study of cardiometabolic risk and obesity in adolescents with Down syndrome, a co-investigator for an NIH Bed-to-Bedside Award, therapeutic targets in African American youth with type 2 diabetes, that is examining ancestry-related differences in treatment response for pediatric type 2 diabetes. Additionally, she is the principal investigator of a 2018 National Institute of Diabetes and Digestive and Kidney Diseases, again an R01 award, studying cardiometabolic risk among South Asian adolescents compared with African American and white peers. Today, Dr. Sheela will be presenting on the topic diabetes and CVD, that is cardiovascular disease risk among South Asians. The next 45 minutes, we will hear more on the subject from Dr. Sheela, and this presentation will be followed by a Q&A session. May I request you all to kindly post your questions in the Q&A chat box, and we'll pick them up after this presentation. So over to you, Dr. Sheela.
Thank you, Priyanka. So hello, everyone, and I want to thank the organizers for inviting me to speak to you this morning on East Coast time and evening for those from abroad. I'm going to be talking today about body composition and implications for cardiometabolic risk in South Asian youth. I hope that in the future we could have a more informal discussion, but I welcome any questions at the end of this presentation. So I'm going to start out by discussing some of the background of the severe consequences of youth onset obesity and the implications of differential adipose or fat deposition on cardiometabolic risk. And I'm using the term cardiometabolic to refer to cardiovascular disease and diabetes. So, and then I'm going to get into some current research regarding ancestry-related differences in body composition that affect cardiometabolic risk and the potential for personalized medicine. So just for those that are not pediatricians, as opposed to in adults where you have discrete BMI cutoffs for the definitions of overweight and obesity, in children who are growing and developing, there are percentile-based distinctions. So a BMI of 85th to 95th percentile for sex and age is how the CDC defines overweight, and a BMI greater than or equal to the 95th percentile for age and sex is the definition for obesity. And here you see the growth curves that we use. On the left is for boys, on the right is for girls. And a BMI at the 5th to 85th percentile is considered normal. 85th to 95th is considered overweight, and greater than or equal to the 95th percentile is considered obese. So in the US, we have seen a significant increase in pediatric obesity over the last several decades. And this has been for a multitude of reasons, decreased physical activity, increase in processed foods. But what you can see here, and I'm just working with, there we go. What you can see here is this increase over the last several decades with an average prevalence of 19% in all age groups, and amongst adolescents of 12 to 19 years old, the prevalence has reached 21%. And this increase in overweight and obesity has occurred all over the world, with increases across the globe. And sometimes this has been attributed also to urbanization in various countries and adoption of a Western style diet. There have been different etiologies in different parts of the world. And in this slide from the CDC, we look at the racial and ethnic distribution of pediatric obesity. Males are here, females here. And what you can see is that in the US, pediatric obesity disproportionately affects our racial and ethnic minorities. So we have a prevalence of 24% in non-Hispanic Black youth and a prevalence of 25.6% in Hispanic youth. So why does it matter? Well, we know from landmark studies such as the Bogalusa heart study that pediatric obesity tracks into adulthood and actually impacts adult health. So this study, part of the Bogalusa heart study, looked at the impact of adolescent onset adult overweight on the prevalence of cardiovascular risk factors. And you can see that they compared an overweight cohort to a lean cohort. And those who had adolescent onset adult overweight had a significantly higher prevalence of hypertension as well as dyslipidemia. However, all adiposity is not the same, and there are implications of fat distribution. And so in this study, the authors are comparing two women, and we're looking at DEXA scans of them. On the left, you have one woman, on the right, you have the other. Both women are matched for age. They're both in their 30s. Their BMIs are in their 30s, approximately 30 kilograms per meter squared, lean body mass of about 44 kilograms, and a fat mass of approximately 31 kilograms. But the authors define the woman on the left as being metabolically healthy versus the woman on the right being metabolically unhealthy based on fasting insulin and the prevalence of insulin resistance. And what you can see is the primary difference is not in the BMI itself, but in the distribution of the adipose tissue. So the woman on the right with the metabolically unhealthy obesity has a central distribution, or what they call android distribution of fat, versus the woman on the left who has more of a gynoid distribution, the lower extremities. And this is similar to, people may have heard of the apple versus the pear distribution, but an android or central adiposity correlates with an increase in insulin resistance and a lot of metabolic abnormalities. And so it's also, the centralized obesity is thought to associate with an increase in what's called visceral adiposity. So visceral fat is the central adiposity that surrounds the internal organs, as opposed to subcutaneous fat. So this distribution of fat has significant implications. So again, on the left, you have what's been termed metabolically healthy obese, and on the right, unhealthy obese. And when you have adipose tissue expansion in the subcutaneous tissue, you have more of adipocyte hyperplasia, which is considered more healthy, versus with visceral fat, you have adipocyte hypertrophy. So you get enlargement of these adipocyte cells. You also have increased inflammatory markers, a decrease in the ability of fat to actually store lipids. And so then you have a deposition of fat in ectopic areas, such as the liver here and in the muscle. And overall, this metabolically unhealthy picture of increased visceral fat and a decrease in the ability to store fat is associated with a decrease in the insulin sensitivity. So in this study out of the Quebec Heart Institute, they're showing you, here you see MRI of the abdomen, and you can see that in this is one individual before and after a 1-year lifestyle intervention. And although the BMI is not very different, 25.9 kilograms per meter squared versus 25.7, You can see the red indicates visceral fat, so that internal kind of pathologic fat. And you do see that after the lifestyle intervention, the visceral fat decreases significantly. And it's 271.4 centimeters squared on the left and 224 on the right. And what you can see here on the bottom left is the body weight, so not a significant difference before and after the lifestyle intervention, but there is a significant decrease of 6 centimeters in the waist circumference. So waist circumference central adiposity has been used as a surrogate measure for visceral fat. Now, how does that impact cardiovascular disease risk? So again, these authors show men on the left, women on the right. Waist circumference is on the y-axis, and the BMI category is BMI is on the x-axis. And in both cases, everyone is obese with a BMI greater than or equal to 30 kilograms per meter squared. But what you can see is the white bars represent a normal lipid, or individuals with normal lipids, whereas the darker bars represent individuals with a high triglyceride and low HDL cholesterol level, which are both consistent with a dyslipidemia pattern. And what you can see is that the increased waist circumference is associated with this dyslipidemia seen versus a lower waist circumference had a normal lipid profile. So there have been kind of 3 scenarios that have been suggested as explaining this relationship between visceral adiposity and the metabolic syndrome. And first, here at the top, you have this increased adipose tissue in a visceral distribution. It's insulin resistant, and therefore you have increased free fatty acids into the portal circulation, causing increased insulin levels, so insulin resistance, glucose intolerance or diabetes, and elevated triglycerides. In addition, in the second scenario, this is also an endocrine tissue. The fat tissue is an endocrine tissue. You have the increase in cytokines such as IL-6, inflammatory factors, TNF alpha. You have a decrease in the favorable adipokine adiponectin, increase in leptin, and you have a pro-hypertensive state. So you have this altered metabolic profile. And then finally, this visceral fat is thought to be dysfunctional fat, or some people have called it an adiposopathy, because the fat is impaired and it has the impaired ability, there's impaired clearance and storage of triglycerides in the subcutaneous fat. So you have ectopic fat deposition in organs such as the liver, the heart, the muscle, and the pancreas. So now I'm going to pivot to some differences in fat distribution by race and ethnicity. And the group from Yale, Liska et al., has compared, they compared 21 white, 17 African-American, and 17 Hispanic adolescents with overweight and obesity. They used MRI to look at the different fat depots. And in this picture, I just want to show you the yellow is representative of the visceral adiposity. The green here is subcutaneous fat, and the subcutaneous fat is divided into the red and the orange, with the orange representing superficial subcutaneous fat and the red representing deep subcutaneous fat, which is thought to be more similar to the visceral fat here. And so they used MRI to look at these fat depots. And what you can see here is they didn't find a significant difference in the subcutaneous fat, but the African-American youth had significantly less visceral fat compared to the white youth and the Hispanic youth. When they divided the subcutaneous into deep and superficial, they didn't see a difference in the deep subcutaneous fat, but the African-American youth had an increase in the superficial subcutaneous fat compared to the white youth. Now, they also use MR spectroscopy to measure hepatic fat fraction. And what's very remarkable here is the African-American group had undetectable, basically had an undetectable hepatic fat fraction compared to the other 2 groups. They finally used MRS to look at intramyocellular lipid as well. And again, here, actually, the Hispanic group had increased fat deposits in the muscle compared to the African-American and the white group. So we're looking at kind of racial ethnic differences in these ectopic fat depots. And here they show kind of three characteristic individuals from their study: the African-American male versus white male and Hispanic male. All had fat percentage 34 to 40%, but you can see that the African-American male has less visceral fat compared to the other two groups and has more subcutaneous fat compared to the other two groups. The Hispanic group has significantly more visceral fat compared to the other two groups. Now, this led me to the thin fat Indian baby paradox. And what this refers to is the finding by Yajnik's group in 2003 originally, and then reproduced multiple times. He found that when he compared white youth from Suffolk, England, white newborns, to Indian newborns from Pune, the white newborns had an increase in birth weight compared to the Indian youth, but the Indian youth had a higher percent body fat. And that's what this is showing. The light color is lean mass and the fat mass in dark. So although the Indian newborns weighed less, they had a higher percent body fat. And interestingly, their mothers also weighed less than their white counterparts. And this finding has been reproduced in multiple immigrant populations as well. So South Asians are the fastest growing major ethnic group in the US, increasing from 3.5 million in 2010 to 5.4 million in 2017. They are known to have greater diabetes and cardiovascular disease risk at relatively lower BMIs compared to those of European and African ancestry. South Asians have higher rates of CVD that cannot be explained by traditional risk factors. And this has led the World Health Organization and the American Diabetes Association to recommend defining overweight as a BMI greater than or equal to 23 kilograms per meter squared for adult Asians, compared to the standard 25 kilograms per meter squared in other adults. So here you see in 2018, the American Heart Association actually put out a scientific statement on atherosclerotic cardiovascular disease in South Asians in the US, discussing epidemiology, risk factors, and treatment, as well as areas for future research. And in the US, here you can see the prevalence of diabetes by ancestral group. So white here is non-Hispanic white. The blue is the Black population, green or teal is Hispanic. This olive green are Native Americans, American Indians, and the gray here are Asian Indians. And they have the highest BMI specific prevalence of type 2 in the US, with a prevalence of 17.4% compared to the other groups. Now, there has been a study, the Masala study, Mediators of Atherosclerosis in South Asians Living in America. I think they have the best acronym. And this is an adult population study of South Asians 40 to 79 years of age. And this was spearheaded from scientists at Northwestern as well as UCSF. And this is a population study looking at with primary outcomes of cardiovascular disease risk, but they also have published a great deal about type 2 diabetes. And what they did in this paper is, in the Masala study, they had 799 participants, and that's this column right here. And they compared their participants in the Masala study to the 4 ethnic groups in the MESA cohort. So that's the white population with 2,600 individuals, African-American with almost 1,900, Latino of about 1,500, and Chinese-American of about 800 as well. And what was remarkable is they looked at the prevalence of type 2 diabetes, and when they adjusted for BMI, smoking, family history, all different site-specific, all kinds of potential confounding variables, the prevalence of diabetes in the South Asian group was 26.7 much higher than any other group. It was followed by the African-American population of 16.4% prevalence, Chinese American 16% prevalence, Latinos of 14.5%, and whites at 6.3%. So a huge burden of disease in the South Asian population. Now, this isn't limited to overweight and obesity, and this is where I think an increase in education for our physicians taking care of the general public is needed in the US. So these authors looked at cardiometabolic abnormalities among normal weight persons from 5 racial and ethnic groups in the US. And what you have on the x-axis is the BMI. On the y-axis is the prevalence of cardiometabolic abnormalities. They define cardiometabolic abnormalities as having at least, so 2 or more of the following: a low HDL, a high triglyceride level, a high fasting glucose level, or hypertension. And what they found, this dotted line here at the bottom represents the white participants. And at a BMI, they had a prevalence of metabolic abnormalities of 30% at a BMI of 25 kilograms per meter squared, right? And so that makes sense given the definition of overweight in some ways in the white population of 25 kilograms per meter squared. However, the next group here are the African-American group. And to have the same prevalence, those individuals had a mean BMI of only 22.9%. For the Hispanic group, they had a prevalence of 30% at a BMI of only 21.5%. The Chinese Americans had a prevalence of 30% at a BMI of 20.9%. And the South Asians had a prevalence of metabolic abnormalities of 30% at a BMI of only 19.6%. So what is a completely normal BMI by US standards. And that's important for healthcare providers to realize. So in spite of having a lower BMI, South Asians have more abdominal fat, so this thin fat Indian, more insulin resistance and hyperinsulinemia, higher levels of inflammation, lower levels of adiponectin or the favorable adipokines, a characteristic dyslipidemia characterized by low HDLs, high triglycerides, and high levels of small dense LDL, and an increased susceptibility to type 2 diabetes and coronary artery disease. So in this study, looking at insulin action, these authors did CT scans to look at fat distribution, and they used euglycemic Indian clamps in 20 healthy Indian American adult males. So the euglycemic Indian clamp is the gold standard for measuring insulin sensitivity. So how well the insulin works to lower glucose. And what you can see here is the Asian Indian compared to 32 African-American and 146 white individuals, adults, and these are males. What I want to point out here is that the body fat, despite the fact that their BMI is around 25, 26 in all 3 groups, actually a little bit lower in the Asian Indian group, the body fat percent was significantly higher in the Asian Indian group at 33% versus 26 and 27%. But the muscle volume was significantly lower. So the Asian Indians actually had a relative sarcopenia compared to the other 2 groups. And in addition, it was only visceral adipose tissue volume that was significantly related to insulin sensitivity. So the visceral adipose, the presence of visceral adiposity explained 38% of the variance in insulin sensitivity. So in sum, the Indian American males had lower insulin sensitivity that was explained 38% in a quantification of 38% by visceral adiposity. Now the Yale group, Jerry Schulman's group at Yale, has also looked at metabolic characteristics of men in different ethnic groups. And this is out of New Haven. And Jerry Schulman's group compared Eastern Asians, Asian Indians, white group, Black group, and Hispanic group. And what I just want to point out here is that the Asian Indian group had a higher fasting insulin level, and HOMA is representative of insulin resistance. So a higher level means more resistant. And this is the insulin sensitivity index. So for this, a higher level is more sensitive. So these individuals, the Asian Indians, had a higher fasting glucose, higher fasting insulin, a higher insulin resistance index, and a decreased insulin sensitivity index compared to the Hispanic, Black, White, and East Asian groups. Moreover, they used MRS to look at fatty liver as well as intramyocellular fat. And they found that the Asian Indians had a higher fatty liver prevalence compared to the other groups, and initially also had increased intramyocellular fat, but this significance disappeared when they adjusted for BMI and age. So we know that South Asian adults have increased percent body fat, increased visceral adiposity, and abdominal fat by CAT scan for a given BMI. They have decreased insulin-mediated glucose disposal, or in other words, decreased insulin sensitivity that was inversely proportional to visceral fat. And they have this increased adipocyte size compared to whites by fat biopsy. And they have evidence of increased liver fat. So we know, so what is the implication of this adipose distribution? We know that after an overnight fast, the abdominal fat releases 50% more free fatty acids to the liver compared to lower body fat. So what does this mean? When you have more central fat, you have an increase here of free fatty acids that are excreted and go into the portal circulation. And the chronic exposure to this adipocyte-derived free fatty acids results in less glucose uptake by the muscle, more glucose secretion by the liver, decrease in insulin secretion by the pancreas, and a decrease in glucose uptake by the fat cells. And so this chronic free fatty acid exposure decreases insulin secretion and may have this beta cell toxic effect. So finally, there are only a few studies that have actually looked at free fatty acid flux in Asian Indians using an oral glucose tolerance test. And these authors back in 2004 did oral glucose tolerance tests on Asian Indians, which are shown by the black dots, and I apologize that it's a little bit difficult to see, compared to white individuals by the white circles here. And what's on the left, on the x-axis, you have the time of the oral glucose tolerance test. Here on the top graph, you have plasma glucose. So with the ingestion of the glucola, the glucose goes up, and then insulin is secreted to bring the glucose down. What is obvious in this graph is that the Asian Indians had this significant hyperinsulinemic response to the glucose. So they had an abundance, a very high insulin secretory rate. But the other thing is that generally the insulin would suppress your free fatty acids, and there was a decrease in this suppression in the Asian Indians. And these graphs are just showing area under the curve. The Asian Indians in darker color, the white here on the right, and the Asian Indians had higher area under the curve for glucose, insulin, as well as fatty acids. So there was decreased free fatty acid suppression by insulin in the Asian Indians compared to white controls. So this led to my current study, which is looking at cardiometabolic health in adolescents of South Asian ancestry, or the CHARISMA study. So the scientific premise of our study is that abdominal and visceral fat distributions in South Asians will lead to an increase in free fatty acid flux, increase in insulin resistance, a decrease in insulin-mediated glucose disposal, and an increase in pancreatic beta cell toxicity. So given the central fat distribution and increased percent body fat in South Asians, we hypothesized that free fatty acid flux may play a more significant role in increasing cardiometabolic risk in South Asians compared to other ancestries. So we set out to compare South Asian American adolescents to African American and white adolescents. So we have 50 youth in each group. Each group is matched. So we recruited South Asians first and then matched African American and white adolescents for age, sex, and BMI percentile. If the BMI percentile was greater than the 99th percentile, we use the raw BMI. Now, in contrast to South Asians, we know from those earlier studies that I mentioned, African Americans have decreased visceral adiposity, and yet they still are at increased risk for cardiovascular and diabetes risk. And so they make an interesting comparison group and important for study. So the aims of our study, our first aim was looking at body composition and free fatty acid flux. So we are using MRI and MR spectroscopy to examine ancestry-related differences in body fat distribution. We are also looking at ancestry-related differences in free fatty acid flux using a 3-hour oral glucose tolerance test, and we're doing mathematical modeling of fatty acid kinetics with the help of Darko Stefanovsky, who's an expert in mathematical modeling at Penn. We're also comparing the relationship of visceral adiposity with free fatty acid flux among the groups, hypothesizing that this relationship is stronger in the South Asian group compared to the other two ancestry groups. Our second aim is looking at glucose and insulin dynamics. We're going to compare beta cell insulin secretory capacity using a type of insulin clamp. And we're going to test for ancestry-related differences in the association between free fatty acid flux and insulin secretory capacity. So again, we're hypothesizing that South Asians have an increase in free fatty acid flux, so it's not suppressed as much by the insulin they're making, and that this free fatty acid flux in turn is beta cell toxic. Our third aim looks at cardiometabolic risk factors, which we are comparing between the three groups. We're going to test for differences in vascular and organ injury using aortic pulse wave velocity between the groups. And the pulse wave velocity looks at the stiffness of the aorta as a surrogate measure for cardiovascular disease risk. And we're testing for ancestry-related differences in the association between free fatty acid flux and these various cardiometabolic risk factors. So this is a cross-sectional study. We're studying adolescents and young adults 12 to 21 with a BMI greater than the 80th percentile for age and sex. And you may remember that I said that by US definitions, overweight starts at 85th percentile. We wanted to go a little bit below that, given that we know that South Asians are at risk at lower BMIs. And then our third inclusion criteria is that the groups should have lived in the US at least since the age of 10 years to take into account the effect of acculturation. Excuse me. Now, South Asian was defined as having at least 3 grandparents identify as being from India, Pakistan, Sri Lanka, Nepal, Bhutan, the Maldives, or Bangladesh. The white group as having 3 or more grandparents identify as Americans of European or white ancestry, and the African American group identify as African American ancestry, that being distinct from recent immigrants from Africa. This study is for the vast majority of participants are being recruited here at Johns Hopkins, and we have an ancillary site at the Children's Hospital of Philadelphia as well, where I'm working with some of my former colleagues from when I was there. We are using MRI and MR spectroscopy to measure visceral, hepatic, and intramyocellular fat. We're doing the 3-hour oral glucose tolerance test and the modeling, the mathematical modeling of the fatty acid kinetics. And we're using this insulin clamp technique to quantify beta cell insulin secretion and the maximal insulin secretory capacity. We're also measuring lipids, lipoprotein particles, adipocytokines, inflammatory factors. We're doing the pulse wave velocity. We're looking at submaximal fitness on a bike test. So unfortunately, South Asians have also been found to have decreased fitness. And so we are looking at that finding, which may go along with their sarcopenia. Anthropometry, including DEXA scans. We're doing 24-hour dietary recalls. We're doing some questions related to acculturation, and we're also doing some exosome microRNA analysis with collaborators at Children's National. So differences in body composition by race and/or ethnicity may have implications for cardiometabolic risk. Studying adolescents permits the detection of early mechanistic and metabolic differences before later exposures may impact the phenotype. And the mechanism of differences in cardiometabolic risk could have implications for treatment. So in the end, why do we do this? My feeling was, if you can find differences in the mechanism, this could lead to a more personalized approach for treatment. So just as an example, if South Asians are found to have an increase in free fatty acid flux, perhaps the first-line treatment should be something that targets that directly, like a thiazolidinedione, as opposed to using metformin as our first line for everybody. And that's just an example, but that's the idea. So I think moving forward, personalized cardiometabolic risk assessment and treatment will be critical. So our study started in 2019, really, but then we did have some roadblocks during the pandemic, since we are recruiting participants to come in. But I have some preliminary results that I was just going to show, and these, are very preliminary. But in this analysis, this was for a scientific meeting last year, where we looked at 14 of the white individuals, 31 South Asians. By design, both groups were similar in age, BMI, and BMI Z-score. You can see that the vast majority are your kind of college age kids who were more ready to volunteer for the study. So far in this analysis, there was not a significant difference in the lipids. There was a higher hemoglobin A1C level, which is a marker of glucose over the last, blood sugars over the last 3 months. It was higher in the South Asian group, although still in a normal range. However, we're also, as I said, looking at body composition. So we then did some preliminary analysis of looking at the different fat depots by MRI. Here you see a comparison, the white group in red, the South Asian group in blue, and these are median values. And you can see there was not a significant difference as of yet, but visceral fat versus seeming to trend higher in South Asians versus than white. But the South Asians did have increase in subcutaneous fat, which was a little bit surprising initially to us compared to the white group. However, then we divided the subcutaneous into the deep subcutaneous and the superficial, which I showed in my earlier diagram. And what we found was there was no significant difference in superficial subcutaneous fat, but there did seem to be an increase in the deep subcutaneous fat in the South Asian group compared to the white group. Again, very preliminary. The total abdominal fat was higher in the South Asian group compared to the white group. Lean mass did seem to be lower, again, with wide confidence intervals, so it's just preliminary. And a higher percent body fat, again, in the South Asian group compared to the white group. We also looked at liver fat using MR spectroscopy, and you can see that the South Asian group did have significantly more liver fat. Now, when you consider what is fatty liver or non-alcoholic fatty liver, generally a liver fat percent of greater than 5 is, or 5 or 5.5%, some investigators use, is consistent with fatty liver. And that's what was used here. So when you use a cutoff point of 5%, whereas none of these white participants had fatty liver, 24% of the South Asians did. So that seemed quite striking to us as a preliminary finding. And when you look at the correlation between liver fat by MRS on the y-axis and BMI on the x-axis, here the South Asians are in blue triangles and the white are in red circles. And I apologize for the switching of colors back and forth. So the South Asians, you can see, had higher liver fat compared for any given BMI compared to the white group. And you just see that at a remarkably lower BMI, South Asians already had significant liver fat. Now, when we started to look, just to review for the non-endocrinologists in the audience, diabetes, type 2 diabetes, you have an increase in adipose tissue causing insulin resistance, and that insulin resistance in the muscle and the liver causes a compensatory hyperinsulinemia. And we think that at some point, the body cannot keep up with the demand. You have an insulin secretory defect, and that's when you get hyperglycemia. But people argue about the relative contributions of insulin resistance versus insulin secretion. So what I wanted to show is, just as a bit of background, in this curve, just keep your eyes on this curve right here. The x-axis represents insulin sensitivity, so how well the insulin works, higher being better. The y-axis is insulin secretion. So if you are on one of these lines, these curved lines, your blood sugar is normal. But what I want you to note is if an individual is sitting here on this line and their insulin sensitivity goes down, so that's the x-axis, goes down, in order to keep their blood sugar normal, their insulin secretion has to go up. Okay, so if you are sitting here and if an individual gains weight and becomes more insulin resistant, their insulin sensitivity goes down, their body will need to make more insulin to keep the blood sugar normal. If they cannot do so, that's when you get progressors to impaired glucose tolerance and even to diabetes. So one of the things we were interested in looking at is, in these different ethnic groups, are there differences in the importance of insulin sensitivity versus secretion? And so in this graph, the blue is white and the red triangles are South Asian. The x-axis is the BMI Z-score. The y-axis, this is a mathematical term, but it's basically a measure of insulin secretion. So again, what you can see is for any given BMI score, the South Asians in red tend to secrete more insulin compared to the white individuals. Now, then we had a small number of African-American individuals. As I said, we're doing the staggered recruitment. When you add in some of their values in green, they seem to have somewhat of a intermediate representation, but again, very preliminary results. Nabatsuda index is a measure of insulin sensitivity. So lower is more resistant. So as it makes sense that as you would get more obese, you would become more resistant. But again, the South Asians seem to be more resistant at lower BMI Z-scores compared to the other two groups. And more recently, we did this analysis of the oral glucose tolerance test. So we went from 0 to 180 minutes. And as I showed you in that earlier diagram, you drink the glucola here, blood sugar goes up, the body makes insulin to decrease the blood sugar down to normal. In this picture, the blue is South Asian and the red is white. And you can see that although there is significant overlap, and this is just preliminary, there seems to be a higher glucose under the curve here. When we looked at insulin secretion, similar to the other study I showed you with the oral glucose tolerance test, we do seem to see this hyperinsulinemic response among the South Asians compared to the white. Now here, I just made this curve. So again, this is that curve I showed you with insulin sensitivity on the x-axis, insulin secretion on the y-axis. And I told you, in order to have a normal glucose tolerance, you have to stay on this curve. But if you are more insulin sensitive, you don't necessarily need to secrete as much insulin. If you are more insulin resistant, you have to secrete more insulin to stay on this curve. And so what you'll note here is the red dots are South Asians, and they tend to be more insulin resistant and secreting more insulin compared to the white participants who are more sensitive and don't have to secrete as much. And so these are just preliminary findings, but this is kind of the direction we're going. And what has been interesting is if you read the literature for adults in India, and the adult studies are generally looking at a much older age group, like 40s to 60s or so. And in those populations, what they seem to show is a decrease in insulin secretion in the adults. So this is not necessarily contradictory to that because we're studying young adults and adolescents. So what we're thinking is a possibility, and we don't know this, but potentially the adolescents could, because they initially have this hyperinsulinemic response and then basically burn out their beta cells and their ability to compensate, which leads to a decrease in insulin secretion later on. But we will see with further studies. So in conclusion, BMI and other clinical thresholds are important tools to assess cardiometabolic risk. However, we have to consider differences in body proportions and body composition due to ancestral racial differences, as well as medical conditions. And differences in body composition can have mechanistic implications, which could potentially lead to more individualized treatment. I want to acknowledge my co-investigators and support through the NIH, as well as Dr. Andrea Kelly, my colleague at CHOP, who is leading our experiments over there, as well as all of our research staff. Thank you. And I'm happy to answer any questions.
All right. Thank you so much, Dr. Sheela. I guess even for possibly non-clinical or non-technical people such as me, we have definitely some interesting takeaways from this presentation. So thank you so much.
I'm glad.
I will hand it over to the audiences. If you have any questions, please feel free to put it in the question and answer box.
Now, Priyanka, should I stop sharing?
Yes, you could. Yes.
Okay.
Thank you.
Yep.
Well, we do have a couple of minutes to take a few questions, Dr. Sheela. In the meantime, I believe while the audiences make up their mind to ask some questions, I did have one question for you.
Sure.
Okay. In the earlier slides, you did mention about the risk or the prevalence of diabetes in the South Asians is relatively higher, and the reasons for which cannot be related to the traditional reasons or causalities. So my first question was, what are those traditional causalities? And number two, have you been able to then sort of list out a few preliminary, list of causalities? If not traditional, then what would those reasons be? In my mind, the traditional, I'm sorry.
Go ahead.
In my mind, the traditional reasons could be lifestyle, socioeconomic reasons, but what else?
Right. So you're right. You're totally right. So traditional risk factors, diet, physical activity, and the real obvious traditional risk factor is BMI, right? Obesity. And when I say that the fact that South Asians have more cardiovascular disease and diabetes at lower BMIs or even normal BMIs is indicative that something else is going on. And a lot of people ask about diet, but when they've done studies in different parts of India, even adjusting for different diet, different cultural practices, still the prevalence of these things is higher. So it's beyond diet. And the genetic factors don't seem to explain a significant proportion either. So in terms of polymorphisms and such. So what I am wondering, and that's kind of the goal of my research, is to find out what is the mechanism. And so in what I presented today, we're wondering if it's this different way of storing fat, more in the visceral or the central portion, and that may have more pathological implications and be more detrimental long-term, if that makes sense.
Right.
Yeah.
It does. Thank you, Dr. Sheela. And one follow-up question to that is more towards the policy and the government intervention, if you will, from that end. So have there been efforts, based on your studies, or based on the your connections within the fraternity and community, to sort of cascade this information with the government stakeholders so that the right kind of policies or right kind of interventions can be proposed within the populations that need to be aware of these.
Right. And I think I definitely have an interest in that, because I find that many primary care providers aren't even aware that Asian adults should be screened at a lower BMI. So a BMI of 23 is considered normal for a white individual, but they should be screened if you are Asian. And so a lot of people are not aware of that. But I showed that picture of the American Heart Association findings. So there is an impetus to increase kind of public health knowledge about the increased risk of different ancestry groups. And I still remember many years ago, which kind of piqued my interest in the early days, of a study of Pakistani taxi drivers in New York, and they did oral glucose tolerance tests and found out that in relatively normal BMI groups, they already had prediabetes and diabetes. And so I think we need to spread the word to screen and to be more aware in the Asian populations, and particularly South Asian, because it does seem to have this different pattern. Even, and I've even noticed amongst my cardiology colleagues, there's a, you may, I don't know, those of us who have gone to their general doctor and had a screening test, they do this risk calculator for your, what's your 5-year risk or whatever, 10-year risk. Well, in the one used in the US, there's no place for South Asian ancestry in that calculation. However, I recently found there's one out of the UK that does take into account being South Asian. And if I've had the experience where if you included that, somebody's risk went up significantly. So I think some of these things are just important for people to learn.
Awesome.
I see that there's a question, let's see, about genetic aspects, family history is collected and correlated. So we are collecting family history of diabetes. And I will say that I do think family history is significant. And just from my own practice in youth onset type 2 diabetes, what I've noticed, for example, in Baltimore, amongst all ethnicities, in my 10-year-olds, for example, who have type 2 diabetes, I find that many of them not only have a family history, it's often in a parent, not just like a grandparent or somebody, but it's a parent. And oftentimes those parents have developed diabetes at a relatively early age, like before age 40. Now, I don't know if that differs by ancestry, but I have noticed that. So type 2 diabetes has almost a 75 or 100% prevalence of family history in a first or second degree relative. So actually, it's more directly genetic related than type 1, which people often don't know. So there is a huge family history component to it.
Awesome. I'm not sure if you already referred to this, Dr. Sheela, but then, when do you expect a more conclusion, the study to conclude and have more final results on this? How long?
Well, so we've completed recruitment of our South Asian group. We're about 2/3 of the way in our white group and about 1/3 of the way in our African-American group. Everything was kind of held up by COVID. So we're hoping in the next couple of years to finish our recruitment. I don't know. I mean, this is a cross-sectional study, so it doesn't imply causation, but I'm hoping that this will lead to further studies to really look at mechanism, not only for South Asians, but also for African Americans, Hispanic. I would love to have a Hispanic group too, because they, like South Asians, seem to have more visceral fat and more fatty liver, but their BMI is not necessarily lower. So I just think that there is room here for a more personalized approach to treat.
Yeah, that's helpful. Thank you. I don't think so. We have any more questions pouring in.
Okay.
Not really. But what I could do is definitely, in case we have some questions after this webinar, I can always shoot an email to you directly and connect you with.
Definitely. And I'm happy offline if anybody wants to contact me. My email is smagge3@jhmi.edu. So I'm very happy to collaborate. I think I was very excited to find out about the India Institute and the work that you're doing. So I welcome any interest.
Awesome. Thank you, Dr. Sheela. It was wonderful. It was actually an excellent speech. I loved it definitely thoroughly. And my eyes were glued to, and pardon me if I've not been able to understand a few technical aspects of it, but then I was definitely trying to take the qualitative aspects as a takeaway. And thanks for your interest. And we'll keep you more posted on the GKII future events, webinars, seminars that we are planning to have. And we'd love to have you on.
Okay. Thank you so much, Priyanka. Take care.
Thank you. Take care. Bye-bye. Bye, everyone.